because it is interruptions or errors in the cell cycle control system that leads to cancer, for example. And so if we can figure out more about how that cell cycle control system works, we could control a lot more disease processes. And we could increase cell growth when we want to. Perhaps we could turn on the cell growth, for example, of nerves in the spinal cord when somebody has an injury. Or we could turn off the cell cycle control system when cells are overgrowing. So there are uses for both purposes, to increase or to decrease cell growth. So that's why the control of the cell cycle control system would be really, really valuable in terms of research. So that's a big area of research. Did you think of any questions you might have about cancer or about issues that are related to mitosis? Come up with any questions overnight? Okay. The second half of the chapter, we're talking about, I thought I said start from this slide, I must have said start at the beginning. Hold on. Okay, so today we're going to talk about meiosis. Some people pronounce this meiosis. I don't know if one is more correct than the other. But to do sexual reproduction, you have to, do you guys know how many chromosomes a human cell normally has? 46. Most of our cells have 46 chromosomes. 23 pairs of chromosomes. Each of you likely, now there are a few anomalies here and there, but most of us inherit 23 chromosomes from our biological mother and 23 chromosomes from our biological father. But we don't just randomly get 23 from each parent. We have to get one of each pair of chromosomes from each parent. It's not like you just throw 46 chromosomes in a hat and you pick out 23 of them. You have to pick out one from each pair. And that's a much more complex issue than just picking half the chromosome. So most of our cells have 46 chromosomes. Our sex cells only have 23 chromosomes, our sperm or egg, whichever we produce. So sexual reproduction depends on both the process of meiosis and the process of fertilization, because it is the process of fertilization that brings back together two pairs of the 23 chromosomes to give the next generation 46 chromosomes. So the first thing we need to talk about is homologous chromosomes. There's a lot of vocabulary. This is one chapter where there's a huge amount of vocabulary. So you need to understand the term homologous. Homo means what? Same. And homologous means the same chromosome. In other words, you have 23 pairs, and each pair are homologous chromosomes. So here's chromosome number 1 you inherited from your biological mother, and here's chromosome number 1 you inherit from your biological father. And when you put them together, that makes a homologous pair of chromosomes. Okay? And then you have chromosome number 2 that you inherited from your biological mother, chromosome number 2 from your biological father, and you have another homologous pair. So what that means is those chromosomes have genes that code for the same thing, the same product. But those genes are not identical to everybody else's chromosome number 1. Now, chromosome number 1 carries maybe 1,000 different genes on it. But your particular version of each of those genes is not necessarily the same as the person sitting next to you. Codes for the same basic product. It may code for hemoglobin. But your hemoglobin and somebody else's hemoglobin are not necessarily totally identical.
Would they be similar or
In identical twins, they would be the same. Now, once you get past birth, well, actually, even once you get past the separation, there are some changes that occur. We found out there's lots of what are called epigenetic changes that happen after fertilization. So identical twins are not. In one sense, yes, their genetics are the same, and it's difficult to distinguish between them. Any of you guys forensic junkies? I mean, like CSI and NCIS and all that kind of stuff, okay? There was a case on the original CSI one time. I think it was the original. It may have been CSI Miami, but anyway, where it turned out, it took them a while to figure it out, but there were identical triplets. Not this one.
Okay.
This one is where there were three young women.
Okay.
They were probably 30, I don't know, something, 35 maybe at this point. And together they planned this murder of one of their husbands. And the way they could finally figure out which one it was because one of them had traveled overseas and had gotten some vaccinations. And so her immune system had some different white blood cells. Everything else was identical, but because identical twins are exposed to different things, there are parts of your immune system that would be different, for example. And so they were able to actually distinguish and figure out which one was the actual murderer. Now, they were all guilty of murder because it was a conspiracy, but to go to trial, they had to know which one committed the murder. So it was kind of a fascinating story. But anyway, there are some differences. But unless you have an identical twin, nobody shares your exact genetic makeup. And to my knowledge, nobody has made a clone, which would also be an identical genetic match. So that's what a homologous pair is. Everybody have a sense of what a homologous pair is now?
Yes.
Okay. So your somatic cells, that's another term you need to learn. Somatic just means body cell. Soma means body. Your somatic cells, all the cells in your body have the 46 chromosomes, except your egg or sperm cells, which only have 23. A karyotype is an arrangement of chromosomes. This is where they collect cells and they treat them with a chemical that stops mitosis, I think, in metaphase. And then you stain them and you match them up. You match them up in pairs. So what you get this array that's just a random mixture of chromosomes, and then you pick out which is pair number 1, which is pair number 2, which is pair number 3. Now in the olden days, we used to do an exercise in lab where we did this, where I would give you an array, and you take paper and scissors, and you cut them apart, and you tried to match up the chromosomes. That was in black and white, it wasn't in color. So it was a challenge to match up these chromosomes. Now we have computer programs that do this. So that's why we don't do it in lab anymore, because nobody does it that way anymore. But what you can do is then match up the homologous pairs of chromosomes. They are matched based on their length and where the centromere is. So the centromere may be close to the middle, or it could be close to the end. There's a couple of them, like this one is closer to the end, this one's closer to the end, this one's closer to the end. This one's right on the edge, right on the end. So the centromere can be anywhere along the chromosome, but that's how they match them up. A karyotype is used in some prenatal diagnosis to determine if there are gross abnormalities in the chromosomes. Now the word gross means what? Does it mean icky?
No.
No. What does it mean?
Large.
Large. It means large. A gross abnormality is an extra chromosome, or a missing chromosome, or an extra piece of a chromosome, okay? Something that you could see on a karyotype. It would not identify like sickle cell anemia where there's a change in a gene. It would only catch really major chromosomal abnormalities. This individual is a normal in terms of number of chromosomes, has the normal number of chromosomes. Is this individual a male or a female? Does anybody know? It's a male. Why is it a male? There's a Y chromosome. In general, and here we're not talking about gender, we're not talking about gender identity, we're not talking about any of that kind of stuff. We're talking purely biological sex. And there's a lot of nuances there. And we've learned a lot of things about how this stuff works. But in general, if there is a Y chromosome, biologically we consider the person male. Okay? Now, things can happen along the way, and male characteristics might not develop or something, or there can be effects on the brain. So it's not an absolute, but in general, that's the assumption. If there's a Y chromosome, it's a male. If there's two Y chromosomes, it's a male. If there's two X chromosomes and a Y chromosome, it's a male. If there's a Y chromosome, it's a male. If there's only X chromosomes, it's a female. Okay, that's kind of the bottom line. But that doesn't answer all the questions, but that's just kind of a starting point. Let's call it the starting point. So X and Y are not technically homologous chromosomes because they do not look alike. They do not code for, they do not have genes for the same products. But we consider them homologous chromosomes for the purpose of this kind of discussion. Okay, so that's just saying. So here's another term you need to know, autosomes. We have, as humans, 22 pairs of autosomes. Autosomes are those pairs 1 through 22, all the way down to 1 through 22. Those are the autosomes. The X and Y are called sex chromosomes. And I've had to change my language because I used to use the word gender instead of sex, and that's not appropriate. So gender and sex are not necessarily the same thing. Okay. So in animals, this is the usual pattern, the life pattern. This is not the same in plants and fungus. It's different. But we think everything does it this way because we do. Okay? So the life cycle of a multicellular organism has different stages. If there is sexual reproduction, there's actually 3 different life cycles. This is the one for animals. So an adult animal has specific cells that undergo meiosis and either produce, ultimately, an egg or a sperm. We call eggs and sperm gametes. You need to know that term, gametes. In general, if you have ovaries, you produce eggs or ova. If you have testicles, you produce sperm. There are people who don't have either one. But in general, that's the pattern. Those cells are what we call haploid. This is another term you need to know. Haploid versus diploid. Haploid sort of sounds like half, right? So haploid have half the normal number of chromosomes. They have one of each pair of chromosomes. So for an animal, only the gamete is haploid. Everything else is going to be diploid. Only the gamete is haploid. Okay? Fertilization takes place. Different animals do it differently. If fertilization takes place, then we have a diploid. That means 2 of each gene, or I'm sorry, 2 of each chromosome. A diploid at this point is called a zygote, fertilized egg. Zygote, fertilized egg mean the same thing. Yes?
Can you say what diploid is again? I'm sorry.
I'm sorry?
Can you say what a diploid is again?
A diploid means you have 2 copies of each chromosome. You have restored the homologous pairs. Now, I try very hard, you may have noticed already, I try very hard to say biological mother and biological father for the next week or so, okay, when we talk about genetics and all that stuff. I know there are lots of ways to make families, okay? Your parents are the people who raised you. But when we're talking about genetics and meiosis and this kind of stuff, we're talking biological parents, okay? Not necessarily the people who raised you, okay?
You said gametes are just like the sexual.
That's, a gamete is an egg or sperm for animals. So a diploid zygote means it has 2 copies of each chromosome, then undergoes mitosis and development until it becomes an adult. So the important point here is that the only time an animal cell is haploid is in the gamete. Okay. And you have to reduce the number of chromosomes so that you can go, when fertilization occurs, you can go back to the full number of chromosomes. If you didn't cut the number of chromosomes in half, then the next generation would have 92 chromosomes, and the next one would have 184. Now plants can do that, but animals generally don't. So humans are diploid organisms, meaning most of our life cycle, we are diploids. We have 2 sets of chromosomes. This is just basically saying what I said in talking about the slide. So anytime you're talking about a sexual life cycle, you're talking about an alternation of diploid and haploid stages. Okay? Sexual reproduction requires this going back and forth between diploid and haploid. So, when do chromosomes duplicate? Interphase. Remember, it's not during mitosis or meiosis. Chromosomes duplicate during interphase. Then, during meiosis, the homologous pairs have to come together so they can separate. We're going to go through the stages here in a second. But they have to come together so they can separate in meiosis 1. And then in meiosis 2, the sister chromatids separate. So meiosis is a 2-step process instead of a, well, you go through the 4 stages twice. In other words, we're going to go through prophase 1, metaphase 1, anaphase 1, telophase 1, and then we're going to go back and go through prophase 2, metaphase 2, anaphase 2, telophase 2.
So, a sister chromatid, is that has 2 chromosomes?
That's your duplicated chromosome.
So
And the 2
one-
chromatids are just the 2 arms or legs, whatever you want to call it.
Oh,
It was the same thing it was in mitosis. It's when the duplicated chromosome is still held together at the centromere. Ask those kinds of questions. Don't sit there feeling confused. I'd much rather you ask the question because other people probably have the same question. Okay? So this is kind of the overview of meiosis. This is kind of what's going to happen in meiosis. Interphase looks pretty much the same. You have G1, you have S, you have G2. Okay, that's your interphase. Now, in prophase 1, I think I've got thought I have a separate slide, I guess not. All right. In meiosis 1, and we use a Roman numeral here. I'm not that picky on that many things, but you've got to learn the correct notation here. We say meiosis 1, and that's a Roman numeral 1. Because for mitosis 2, we use the Roman numeral 2. Okay? We have prophase 1, metaphase 1, anaphase 1, telophase 1. Okay? In prophase 1, The same four things happen that happened in prophase of mitosis. What were those four things? The centrosomes or centrioles separate. The nuclear envelope goes away, sort of disappears. What? What? The spindle fibers form. What? The chromosomes condense. Okay, those four things happen. The centrosomes or centrioles separate. The mitotic spindle fibers or the spindle fibers form. The chromosomes condense, and the nuclear envelope disappears. Okay, those same four things happen in prophase 1, but we have two more things that happen. Two additional things happen in prophase 1. One of those things has the name, somebody got into my numbers. Is called synapses. Synapses. The process of synapses is when chromosome number 1 that you inherited, duplicated chromosome number 1 that you inherited from your biological mother, and duplicated chromosome number 1 that you inherit from your biological father find each other and pair up. Yes? This is in prophase 1. We call this process synapsis. It's when the homologous pairs come together. The structure that they create is called a tetrad. What does tetra mean in Latin or whatever? 4. Tetra means 4. A tetrapod has got 4 limbs, okay? We are tetrapods. 2 arms, 2 legs. Okay, so the tetrad comes from the fact that there's 4 sister chromatids. Now, the other thing that happens in prophase 1 is crossing over. Now, we're going to come back and talk about crossing over. So you may want to leave a space there, or you may want to say, just write crossing over and then say see ahead or something, okay? But a lot of stuff happens in prophase 1. In metaphase 1, the homologous pairs line up. Instead of all 46 chromosomes lining up on that plane, remember if you cut the styrofoam ball in half and you have 46 chromosomes there, instead of that you have 23 pairs of chromosomes that line up there. One of each pair is going to be above the plane and one of each pair is going to be below the plane. So they're going to be like on top of each other. If you watch the video, I don't know how to get the sound on the video yet, but if you watch the video, it'll show you that in much better detail. The other thing that happens then in metaphase is something we call independent assortment. Independent assortment. And again, we're going to talk about that in a minute. But that happens during metaphase 1. In anaphase 1, instead of separating the sister chromatids, we separate the homologous pairs of chromosomes. So chromosome number 1 you inherited from your biological father goes one direction, and chromosome number 1 that you inherited from your biological mother goes the other direction. But they're still duplicated. Notice here, instead of having 2 little tails hanging behind, now we actually have 4 little tails hanging behind. Yes?
So the parents
No, the pairs are separated from each other.
So the pairs are broken apart.
Right, this is chromosome number 1 from the biological father. This is chromosome number 1 from the biological mother. This is chromosome 2 from the biological father. This is chromosome 2 from the biological mother. So the pairs separate. Okay? Other questions? Okay. In telophase 1, and usually cytokinesis, the nuclear envelope reforms and generally cytokinesis takes place. The chromosomes may or may not uncondense. They may partially uncondense. So now we have 2 haploid cells. So in meiosis 1, we have cut in half the number of chromosomes in each cell. We started with 4 chromosomes, 1, 2, 3, 4 chromosomes in our parent cell. And our daughter cells are each going to have 2 chromosomes. In fact, the word meiosis means reduction, I think in Greek. Okay? Now, meiosis 2 is almost identical to mitosis. The biggest difference is you have 2 cells going through it. Instead of just 1 cell going through mitosis, you have 2 cells going through meiosis 2. The nuclear envelope disappears, the centrosomes separate, the mitotic fibers, the spindle fibers form, the chromosomes condense if they had uncondensed in 2 different cells. In metaphase 2, the chromosomes line up along the equator. In anaphase, the sister chromatids separate. And this is the big thing that I had a hard time understanding when I was sitting in your seat. Okay? In anaphase 1, in anaphase 1, homologous chromosomes separate. In anaphase 2, sister chromatids separate. Okay, let me say that again. In anaphase 1, homologous chromosomes separate. In anaphase 2, sister chromatids separate. So if you had a test question that said, when do sister chromatids separate? And it was, a couple of wrong answers, and then anaphase 1 and anaphase 2, or anaphase 2 and anaphase mitosis, what would be the right answer? Anaphase 2 and anaphase mitosis. So those are the kinds of things, you've got to be able to put this information together. So that's what I'm saying. Meiosis 2 is almost the same as mitosis. Yes?
So before meiosis 2 begins, it doesn't go back to interphase, right?
They call it has a name, but it's not really interphase. Don't worry about that at this point. The chromosomes don't duplicate again or anything like that. So just for our purposes, even though it's not absolutely correct, for our purposes, just kind of assume we go straight from prophase 1 from telophase 1 to prophase 2.
So the only difference between meiosis and mitosis.
Say that.
The only difference between meiosis 3 and mitosis is just that meiosis is 3 cells opposed to 4.
The difference between meiosis 2 and mitosis, or meiosis and mitosis?
Meiosis 3.
Okay, the difference between meiosis 2 and mitosis is you'd always deal, in meiosis 2, you're always dealing with a haploid cell. And in mitosis, you could have either a haploid or a diploid. And you generally have 2 cells going through it. In meiosis 2, you generally have 2 cells rather than 1. then telophase 2 and cytokinesis, again, the nuclear envelope reforms, the chromosomes decondense or uncondense, and usually you have cytokinesis, usually the cell divides. And this is primarily what happens, this is more accurately depicting what happens in animal cells than in plant cells. So here's a little bit of a comparison, yes.
So does meiosis only happen when you're like in embryo?
Does meiosis only happen in the embryo? No. In human females, and this gets really complicated, human females are born with all their eggs they're ever going to make are like frozen in metaphase 1, I think, of meiosis. Men, however, they do meiosis all the time. Never stops. So, meiosis in women is fairly complicated. One egg per cycle then ripens and goes through the rest of meiosis 1, but meiosis 2 doesn't actually complete, I think, until fertilization or until right at fertilization or something like that. So it's more complicated than women are just more complicated, which is cool. So, if you're interested in development, I recommend that you take developmental biology with Dr. Livingston at some point. It's offered every other spring, I think. And it's an amazing course. It doesn't just focus on development of humans. It's a developmental biology course. It is pretty cool. So this is a comparison of mitosis and meiosis that's kind of useful. There are many ways to study. One way to study, remember any of you Sesame Street kiddos? Did you watch Sesame Street when you were little? We were really not a Sesame Street family. My kids didn't like it that much. But I do remember they used to have this little game they did, and one of these things is not like the other, you know, and they'd have an apple and a banana and an orange, and then they'd have a toy giraffe or something, you know. And you play one of these things is not like the other. Learning what is the same and what is different is an excellent way to study. What do these things have in common? How are these things different? It's a very good way to organize things in your head. Okay? So this is just a comparison. All right. Genetic diversity. Diversity is a buzzword. It's becoming more and more and more of a buzzword. Some people are uncomfortable with the word diversity, but diversity is a good thing. From a biological perspective, why is diversity a good thing? Why is it a good thing that we're all different?
So we can evolve.
For evolution, why is that important?
Survive.
It's important for survival of the species. And this is where it becomes a little bit dicey for some people. Diversity is not necessarily good for the individual, but it's really good for the species. Okay? If somebody walks in this room right now and sprays this room with smallpox virus, chances are pretty good I'm the only one that has been vaccinated for smallpox, because they stopped doing smallpox vaccinations back in the 70s. And my dad got one of the last ones. He was going out of the country, and they gave him a booster kind of smallpox vaccination. But they stopped routine vaccination for smallpox in the 70s. And it's the only disease the CDC and the World Health Organization has actually eliminated from Earth, through vaccination, by the way. And so my immunity might or might not still be really good. But if somebody came in here and sprayed this room with smallpox, would all of you get smallpox? No. Even though you haven't been vaccinated against it, not all of you would get smallpox. Probably half. Now, of those half that got smallpox, let's say this side of the room gets smallpox. Congratulations. Would you all die? Even I'm talking pre-medical intervention, without going to the ICU or something. Would all of you die? No. Why not? About half of you would die.
Because we have antibodies that other people don't.
Yeah, because your immune systems are different, you've been exposed to different things, you have different genetics, and for purposes of survival of the species, it's a really good thing that only 25% of this class would die from smallpox. But if someone came in and sprayed another deadly disease, a different 25% might die. So it's good to have diversity. Now, if you came in here and sprayed us all with Ebola, what would probably happen? Probably all of us, maybe one would survive. And again, without any medical intervention, even that would be a small probability, okay? Because we did not co-evolve with Ebola. We don't have any immunity. We don't have any historic immunity to Ebola. So, diversity is a good thing for a population, for a species, okay? So, nature has done things to increase diversity. I guarantee you on the next test, one of the questions, one of the short answer questions, is going to be list and describe the 4 contributors to genetic diversity. Okay? List and describe the 4 contributors to genetic diversity. And I have them listed on the PowerPoint in color. Okay? If you're colorblind, I'm really sorry, but do the best I can. Okay? So here, the number 1 source of genetic variability is mutations. Mutations are actual changes in DNA sequences. They happen randomly. You cannot predict what genetic mutation is going to take place. A genetic mutation might be beneficial, it might not be beneficial. It's random. All organisms have mutations. When you have sexual reproduction, there are 3 more contributors to genetic diversity. So mutation is kind of the first. The additional 3 include independent assortment. This takes place in metaphase 1 of meiosis. Independent, what does the word independent mean? Doesn't depend on what anybody else does, right? Assortment, what does assortment mean? It can mean a lot of different things. What?
Like arrangement.
It's an arrangement, it's how you put things together. So independent assortment in this context is determined by how the chromosomes line up during metaphase 1. If you have 2 pairs of chromosomes, they could line up, we're just going to be stereotypical here, the blue ones are going to be from the biological father and the red ones are going to be from the biological mother, okay? So, chromosome number 1 from the biological father and chromosome number 2 from the biological father happen to be on the same side, and from the mother happen to be on the other side, so that when they sort, when they go through then anaphase, both the ones from the father are one side and both the ones from the mother on the other side. So that's one way to do it. Possibility number 2, when you have 2 pairs of chromosomes, is number 1 from the father and number 2 from the mother are on the same side. That's a little hard to see when you only have 2 pairs of chromosomes, but you end up with 4 different possible gametes, basically. So here's a picture where you have 3 chromosomes. And there are 4 different ways you can line up those chromosomes. You can put, if we choose to make the father the white ones and the mother the black ones here, you could have all the fathers on one side, all the mothers on the other. You could have number 1 for the father with number 2 and 3. No, wait a minute, I did that back. Number 1 for the mother and number 2 and 3 from the father together. You can have 1 and 2 from the father, from the mother, and 1, 3 from the father together. Or you can have 1 and 3 from the father and 2 from the mother. So there's 4 different ways you can line them up, which means you get 8 different possibilities for your gametes. Well, it turns out that the formula for how many different gametes you can get is n is 2 to the n power, where n equals the number of pairs of chromosomes. Okay? So if you have 2 pairs of chromosomes, 2 to the n, or 2 squared would be, 2 squared. Squared is 4, right? So if I go back, here I get 1, 2, 3, 4 different possibilities, right? If n is 3, 2 to the 3rd power is what? 8. All right? 2 to the 4th power would be 16. 2 to the 5th power is 32. Well, how many pairs of chromosomes do humans have? 23. 2 to the 23rd power is almost 8.4 million. It's 8.3 something million. Let's put that in perspective. How many eggs does the average woman produce in her lifetime? Mature eggs does the average woman produce in her lifetime? Let's think about it. Let's assume the onset of menses at age 12. We'll pick easy numbers. And we'll say the onset of menopause is 52. So how many years is that? 40 years. And how many cycles per year? It's actually 13. Every 28 days makes 13 cycles a year. So 13 times 40 is how many total cycles? 520. So women in general, if they never get pregnant and they produce 1 per cycle, 1 mature egg per cycle, would produce about 520 eggs in their lifetime. What are the chances of any 2 of those eggs being identical? Well, it would be 520 in 8.4 million. Pretty small odds, okay? Now, what are the odds of a man producing the same sperm? Well, how many sperm do men produce? Billions and billions, okay? They produce millions every day. So what are the odds that a man will produce the same 2 sperm? That's pretty good over the lifetime, okay? So, but then when you add into that what we call random fertilization, and this can actually mean 2 different things. It can mean which pollen granule gets to which flower is random. It can also mean which sperm gets to which egg is random. So even if you have a monogamous couple who have a chance of producing, in theory, well, they don't really have a chance of producing that many but anyway, if a woman produces 520 eggs in her lifetime and a man is producing, millions of sperm a day, the chances of a specific sperm meeting a specific egg would be 8.4 million times 8.4 million, which is something like 7 times 10 to the 13th or something. Way more people than have ever existed. So, no, you're not going to get 2 identical people that are not identical twins. Primarily because a woman produces too not enough eggs to allow that to happen. A woman would have to be producing millions of eggs and be monogamous with one man to even come close to predicting that there would be another human that would be the same because of independent assortment. So independent assortment gives us a huge amount of genetic variability. And that's why, anybody ever do the 23andMe or what's the other one? Ancestry.com, where you send in your spit or and you may be startled to find that, first of all, you may find out something you didn't want to know. They've actually captured criminals that way because they, well, who was it? The guy in California who was wasn't the Hillside Strangler, he had a name, he was a serial murderer. He didn't send his DNA into 23andMe or whatever, but one of his relatives did. And it was a close enough match that they knew that the killer was a relative of this person. And so they systematically looked at all the male relatives of this person, and they eventually identified this guy. And it brought up all kinds of legal issues. But anyway, so be careful when you do stuff like that. But you can have full siblings have the same parents, and they can come up with different percentages of being, from different parts of the world, because it just depends what you happen to inherit. And I have two sons. They could have inherited different things. Now we're pretty boring. Anybody have to do their census or help their parents do their census? And this time they wanted to know what your ethnic background was. It's like, my kids are calling me and it's like, I don't know, English, Welsh, Scottish, Irish. we're pretty boring. I mean, we're just, we don't have anything exciting.
Would twins, like, for girls, would they have, like, more closer, like, the same background, like?
Fraternal twins would be no closer than any siblings. Now, identical twins should have come up the same. And my husband was defending it. He said, well, they sent identical twins to different labs. And it's like, well, yeah, they have the same genetics. But siblings who have the same parents are not necessarily. Each characteristic, they have a 1 in 4 chance of having the same thing as their sibling. So the combination is not going to come out the same.
It's not, is it possible to be identical with different genders?
No. Because you wouldn't be identical, you wouldn't have the same chromosomes.
Because the reason I'm asking that question is because, like, I have friends who say, like, my brother, me and my twin brother, we're, like, both, like, we got, like, two twins, so everybody else is, like, really dark, and my kids are always like, oh, you guys got the genetics from, like, Indian or whatever. But then when she asked that question, she was like, are they identical to you? That's so fun.
Yeah.
My twin brother, like, can I see a picture? You guys are identical? I'm like, no. They're like, thank God. We're all.
But I mean, the fact that fraternal twins could be more similar than their siblings, that's just luck of the draw.
Yeah.
I know fraternal twins who are extremely different. And there have been cases where the mother is, there's one in England where the mother is from like Nigeria and the father is, I don't know, Norwegian or something. He's as light-skinned as you can get and the mother is as dark-skinned as you can get. And they have twin girls, and one appears more African and one appears Scandinavian.
We're down with magazines and stuff.
Yeah, but they're full siblings. Just what you happen to inherit.
Is it possible for, like, two darker skinned people to have a lighter skin?
Yes. We'll talk about that in the next 1 or 2 chapters. And two lighter skinned people can have a darker skinned baby. Not hugely different, but definitely different. So random fertilization is a big contributor to genetic diversity. The fourth thing that's a contributor to genetic diversity is crossing over. This happens in prophase 1. And this is an amazing process if you think about it. When synapsis happens and the tetrad forms, those pairs of homologous chromosomes are very close together. And this is where we use the term non-sister chromatids. This chromatid right here, this blue one, and this red one right next to it, those are non-sister chromatids. They will cross over, they will physically cross over one another. And sometimes they will exchange material. The place that happens is called a chiasm. I'm not going to ask you to know that. The site where that happens is called a chiasm. And you end up with down here, after you get through the whole process of meiosis, you end up with a chromosome that never existed before. This is a whole new chromosome. It did not exist in either the mother or the father. It is a recombination of chromosomes. And when that happens, it always happens between genes. It never like cuts a gene in half. It always happens between genes. There's something wrong with the fire alarm system. It's going off Sunday. And it's always the same number of DNA pairs that are traded. So if there's 500 on this side, there's 500 on that side. Okay? Are other people leaving?
Yeah.
All right, pack up and leave. I will